Module 1, Part 2, Light Microscopy. We will discuss the general principles of light microscopy in this part. The average dimension of typical animal cell is 10 to 30 microns in diameter, which is 5 times smaller than the smallest particle visible to naked eye. The first good microscope was developed in the 19th century. At that time, it was discovered first that plant and animal tissues are actually aggregates of individual cells. This idea or discovery is known as cell doctrine and marks the birth of cell biology. Early microscopes like this one was used by Robert Hooke in 1660s, used the lenses or a mirror to illuminate the specimen and first revealed the existence of cells, which revolutionized science. Development of optics and light microscope during the 19th century led to the mid-20th century highly sophisticated microscopes limited only by the resolution of light. In the second half of the 20th century, the fluorescence microscopy, confocal techniques, and the recent past, several super-resolution microscopic techniques pushed the light microscopic resolution into the nanometer scale previously thought impossible without an electron microscope. So if you ask the typical person, what is the most difficult aspect of observing small cells? The most common answer is the magnification and is the major challenge for observing small structures. There are, however, some other challenges to consider. Another is resolution, and the third is the proper sample preparation. So we will take a look at each of these challenges individually. Magnification is the function of the microscope the scientist uses, and in particular, it is the function of one specific lens, that is the objective lens. Here is a figure of a setup of a simple light microscope or bright field microscope. The red line has been added to show the light path through the microscope. So there is a light source, a lamp, and a series of collector lenses, a mirror, condenser, focus the light on the specimen. I want to mention here that specimens for light microscopy are usually mounted on a clear glass or plastic slides or clear glass or plastic dishes, and they sit on a stage which is usually adjustable. So once the light passes through the specimen, it is collected by the objective lens, and again, it is the objective lens which provides most of the magnification in the light microscopy. In this example, the objective lens is 100x or 100-fold magnification. The most simple light microscopes have adjustable objective lenses, which can be 4x, 10x, 40x. These type of microscopes and objective lenses are probably the type you are most familiar. So the light passes through the objective lens and then through the projection lens, which often gives additional magnification. And in this example, it is 10x, which is usually common to most light microscopes. So in this example, the final magnification for the light microscope is 1000x. From the projection lens, the light passes to a detector. And today, the detector can be a computer screen, but most often the detector is just your eye. So that was magnification. So now let's talk about resolution. A very simple definition of resolution is the ability to distinguish between two objects. All microscopy depends on beams of radiation such as lights or electrons, and if you will remember from physics classes, the beams of radiation travel in waves. And the wavelength of a beam of radiation is denoted by the Greek letter lambda. The wavelength is the distance in between the two valleys or two peaks of the wave. The resolution depends on wavelength of beam of radiation you are using. A beam cannot be used to probe structures smaller than the wavelength. So light microscopy limits are set by the wavelengths of a visible light, which is in the range of zero point 0.4 microns for violet to 0.7 microns deep red light. So light microscopy cannot be used to probe structure with a distance of less than 0.45 microns between them. I will elaborate so you can fully understand that concept. Resolution is denoted by the letter D, and here is a second definition so you can understand the concept. Resolution is the minimum distance between the two distinguishable objects. For example, if your microscope system has a resolution of 0.5 microns, it means you can never distinguish objects less than 0.5 microns apart. If less, then objects will appear as one. So how do you calculate resolution? Here is a formula for resolution. The resolution is directly proportional to the 0.61 lambda or wavelength, where the 0.61 is actually a constant in this formula. The resolution is inversely proportional to the value of the numerical aperture, or n sine alpha. Here, the n is the refractive index of the medium the specimen is sitting in. Under the microscope, all specimens are in some type of medium, air, oil, or even water. And all of the media have a refractive index, which is represented by the n in this formula. The alpha is the angular aperture of the microscope system you are using. So to understand, let's look at the simple diagram of bright field microscope. So you can see that light is collected by a condenser lens focused onto the specimen, and the light passes through the specimen and collected by the objective lens and projected onto a detector. When the light hits the specimen, it hits at a particular angle, and that angle is 2 alpha. So alpha is half of that angle. So let's do a simple calculation. So what is the best resolution you can obtain from a simple bright field microscope? So best maximum angular aperture you can get today is about 70 degrees. As previously mentioned, the shortest wavelength of light is 0.45 microns, and the best medium is immersion oil with a refractive index 1.5. So if you put all the values into the formula, the result is 0.194 microns or micrometer, or approximately 0.2 microns for a simple light microscope. Anything smaller than 0.2 microns will appear as one object when conducting simple light microscopy. Here is a sample image of the bright field microscopy. The image is good. You can see the image of the cell and some inner detail, and you may make out some organelles. There are some advantages and disadvantages to simple bright field microscopy. The advantages are simple and inexpensive. Microscopes are readily available and not cost prohibitive. You do not have to stain the sample to see some kind of image, and you can image live cells because there is minimum manipulation of the sample, so you can keep the cells alive. But there are some disadvantages or limitations. There is low contrast of most biological samples because most biological samples are composed of primarily of water, which makes for low contrast in the specimens. As discussed previously, there is low resolution. 0.2 microns is the best that can be achieved, which is good, but you can do better. Another problem with the light microscopy is that you are collecting light from the plane of focus and collecting spurious light from outside the plane of focus, which blurs the resolution or make the resolution worse. But there are some optical, physical, and biochemical ways to minimize these limitations. Let's look at some of the optical methods to improve the light microscopy. One of the ways is to use different types of light microscope. Several different types have been invented, and it is the physics of the light microscopes that make them differ from the standard light microscopes. I won't go into the physics of the various scopes, but will mention a few differences. In the bright field microscopy, light from a tungsten lamp is focused on the specimen, usually stained with a dye to enhance the contrast by a condenser lens below the stage. In a phase contrast microscopy, which increases the contrast of biological specimen, the incident light passing through an annular diaphragm focuses a circular annulus or ring of light onto the specimen. Light passing unobstructed through the specimen is focused by the objective lens into the thicker gray ring of the face plate, which absorbs some of the direct light and alters its phase by 1/4 of a wavelength. If a specimen refracts or bends or diffracts the light because of the refractive index of the material, the phase of some of the light wave is altered, which is shown by the green lines. And the light waves pass through the clear region of the faceplate. The refracted and unrefracted light is recombined at the image plane to form the image. Let's compare the images from these microscopes. The first variation is a phase contrast light microscope. As mentioned previously, the medium that sample is in has a typical refractive index, but all of the different components of the cell have been different refractive index depending on their makeup, so the nucleus has a little bit of different refractive index. The vesicles have a different refractive index, Golgi apparatus, etc. So phase contrast microscopy take advantage of these differences when the light is projected onto the sample, a series of annular rings, and depending on the refractive index of the inner cellular components, it will push the light out of phase or keep the light in phase. Light out of phase is dark and light in phase appears very light. So the result is you can see more details inside the cell. So see the comparison of the bright field microscope versus the phase contrast image. You can appreciate the detail gained at the intracellular level. The phase contrast microscopy capitalizes on different refractive indices of cellular components and gives greater detail of the organelles. In contrast, you can see the differential interference contrast, or DIC, or Nomarski microscopy. DIC, which relies on polarized light, produce a very different image, but very useful. With DIC, you can appreciate that image is almost a 3D image or pseudo image of the cell. So you can gain the same information about the three-dimensional structure or characteristic of the cell. So those are some optical methods to improve light microscopy. What are some physical and biochemical methods? So now we can look at the third challenge. How do you handle and prepare your samples before microscopy? Good sample preparation can actually improve on simple light microscopy. Sample preparation occurs in 4 steps. First is the fixation step. Second is embedding. The embedding and sectioning steps are for thick specimens only, like tissue samples. Cells growing in culture do not require these 2 steps and can be directly fixed and stained on the slides. Let's take a look at them individually. So start with fixation. So live cells are in a dynamic state. The organelles are moving, the vesicles are being trafficked, motile cells such as macrophages, neutrophils are on the go, lower eukaryotic cells are motile. All the movement complicates microscopy because as the cells are moving around, resolution is lost and the image is blurred. So fixation takes care of that by actually freezing the cells and stops all that movement. It also kills them, which may be good or bad. Fixation freezes the cells in time, not literally freezes in ice or something, and stops all the motion so you can capture a good image. The way it freezes is by cross-linking macromolecules. The agents used for cross-linking are glutaraldehyde and formaldehyde, which form covalent bonds between the free amino group in proteins. Fixation also acts to partially permeabilize the cell, so if you are interested in staining your cells or tissue specimen, fixing facilitates that by permeabilizing the specimens. The second step in the sample preparation is sectioning and applies only to the thick specimens. If you are looking at a single layer of cells, then sectioning is not required. But if you are looking at a tissue specimen from plants or animals, then some sectioning is necessary. Most biological samples are too thick to allow light to pass through, and if light can't pass through them, you cannot conduct light microscopy. You have to slice through or section the tissues into very thin sections so light can pass through so you can capture an image on the detector. Most biological tissue samples are too soft or flimsy to section, so you have to fortify or embed the sample. To do this, first you put your sample into some kind of liquid embedding material such as molten wax. Most commonly now, you put the sample into a liquid polymer or plastic, and after cooling of wax or polymerization of the plastic, you end up with a solid block containing the specimen to be sectioned. Then you place the solid block with the specimen embedding in the center of the block and put it on a sectioning apparatus known as a microtome. You can think of it as a similar to a slicer used in a deli. Place the block on the arm of the microtome and the arm will go up and down and pass the block over a very sharp knife, sometimes a diamond knife. And you will slice the specimen into very thin sections and can be placed on the microscopic slide for light microscopy. The third step in the sample preparation is staining. There are a number of histological stains, dozens and dozens that are available, all of which enhance the contrast certain structure within the cells. I want to show you a few that are used commonly. Hematoxylin is very common. It is a nuclear stain and stains the nuclei of the cells in a blue-violet color. It is a base and is attracted to acids. So it binds, for example, to the DNA, which is an acidic molecule, and highlight the nuclei in the cell. Another stain is eosin. It is an acid which binds to the basic macromolecules, and many cytoplasmic proteins are basic. So it colors the cytoplasm pink. These two stains are often used together and commonly known as H&E stain. See an example of a stained section of gastric mucosal body showing the parietal cells by light microscopy. Please note that in your textbook, the 8th edition of your textbook has a print error saying exactly the opposite. This information is correct. Hematoxylin is a basic stain, eosin is an acidic stain, but this information is just reversed in your textbook, so just be careful. This is the correct information. Peripheral blood smears are commonly stained with right giemsa stain, which is a combination of eosin and methylene blue. The acidic nuclei stain is blue and the cytoplasm is pale pink. The red blood cells or the erythrocytes which lack the nuclei stain a pale pink. These smears are used to identify and count white blood cells. Another commonly used stain is benzidine, which is a stain that binds heme-containing proteins. So you know blood cells contain the heme. This slide is a stage 35 xenopus embryo stained with benzidine, and you can see the ventral blood island stained very well with this stain. This concludes part 2 of module 1.